On volume? Sounds like we are. Thank you, Dr. Wright. My name is student Dr. Josh Beckler. I'm a first-year medical student here at KCU, and I actually graduated from the College of Biosciences in May with my master's degree in biomedical science. So in the time frame between graduation in May and then matriculating into the first-year class in August, I was able to take part of the Summer Student Research Fellowship through the KCU Office of Research, working in Dr. White's lab, specifically with Miss Megan Woods right over there. She's a co-author on this work, and ultimately this is what we were able to produce. So I understand that I'm the last thing standing between everyone and lunch, so without further ado, this is a potential therapy for DMD, specifically looking at cardiomyopathy in Dmd model mice. First, let's lay some groundwork on the disease of concern, and that's Duchenne muscular dystrophy, which I will refer to as a DMD for the remainder of the talk. DMD is caused by an X-linked recessive mutation in the gene coding for the dystrophin protein. It has an incidence that roughly occurs in every 1 in every 3,500 live male births, and this makes this one of the most common inherited neuromuscular disease in boys. It has a lethal and very severe disease outcome with a devastating course for these patients. They experience progressive muscle degeneration disease. These signs and symptoms are typically noted by as early as the second year of life. They can be wheelchair bound by the age of 12, and then the lifespan is ultimately cut short in the third decade of life, and this is typically due to cardiac or respiratory failure. One important thing that we want to remember when we're talking about DMD is that as it stands right now, there is no cure for this disease. Therapy for these patients is strictly palliative. So the project that myself and Megan Woods worked on specifically looked at cardiomyopathy in DMD. The reason why this is important is because of the role that cardiomyopathy plays in the later stages of the disease. So it's been observed that the incidence of dilated cardiomyopathy increases during the teenage years, and by the end of the second decade of life, nearly all DMD patients will indeed show some sort of signs and symptoms of cardiomyopathy. Ultimately, cardiomyopathy alone will count for up to 40% of all the deaths in these DMD patients. So Dr. White's lab has been investigating a new therapy for these DMD patients, and that's expression of a dystrophin isoform known as retinol dystrophin. And we'll talk about that here in a little bit, in a place where it's not normally expressed, and that's the muscle tissue. So our work specifically looked at the analysis of transgene therapy on cardiomyopathy in Dmd model mice. The dystrophin protein itself is a very large protein weighing in at about 427 kilodaltons. It's found in striated muscle, so of course that includes skeletal muscle as well as cardiac tissue. And as you can see by the image here, this is from a Nature review, its role in muscle cells is to link the actin cytoskeleton of the cell to the extracellular matrix, specifically the basement membrane. And it does this via associating with a number of integral membrane proteins that ultimately make up the dystrophin-associated protein complex. Key takeaway point here is that dystrophin is a structural protein found in muscle cells, and it's key for their integrity, particularly in the active contraction and relaxation phases. There are a number of dystrophin isoforms, however, and the one that we're most interested in is that of retinol dystrophin. It also goes by the name of DP260 for its molecular weight of 260 kilodaltons. The way that retinol dystrophin is generated is via alternative splicing of the dystrophin gene. So the dystrophin gene is a very large gene, as many of you know. It's actually 79 exons. So retinol dystrophin is lacking exons 1 to 29. It starts at exon 30. The important feature about this is that a subset of the DMD population, approximately 1/3 of all these patients, have their mutations in the dystrophin gene that is upstream of exon 30. Therefore, they are able and are actively producing retinol dystrophin in the eye, however, not in their muscle. The other unique feature about this isoform is that it contains 3 of the original structural domains. And most importantly, it can still bind actin, and it can also associate with the dystrophin-associated protein complex. Last thing I want to point out on this slide is that on the right we have a Western blot, and when we probe for dystrophin, in muscle we pick up a single isoform that comes in at 427 kilodaltons. This is our full-length dystrophin protein. When we look at retina, we're once again picking up that DP427 full-length dystrophin isoform. But now we're also picking up a band at 260 kilodaltons. That is our retinol dystrophin. So retinol dystrophin, protein that may be able to assume the same function as dystrophin in muscle, but typically only expressed in the eye. So in order to investigate the efficacy of this therapy, we turn to a DMD mouse model that has this very severe dystrophy phenotype. The reason that's important is because it closely mirrors the clinical outcome in human patients. These mice exhibit progressive muscle degeneration. They have loss of mobility in the limbs, particularly the hind limbs that come along with gait disturbances. Abnormal spinal curvature, particularly when the paravertebral muscles are affected, and then ultimately early death. So naturally, the question that we've been building up to here is, what is the outcome of expressing this dystrophin isoform, retinal dystrophin, in the place where it's not normally expressed in the muscles of these DMD model mice? Would it be able to rescue the severe phenotype that they exhibit? So in order to evaluate this, a transgene was created that allowed the expression of retinal dystrophin, human retinal dystrophin specifically, in the muscles of these DMD model mice. And these were the results. So important thing to remember in the next few images I'm going to show you is that both of these mice are DMD model mice. Neither of them is able to produce full-length dystrophin protein. On the right, we have the example of a severe example of a DMD model mice. You can see the reduction in growth. This mouse was pretty much unable to use its hind limbs and ultimately had a lifespan that was terminated at 3 months of age. On the left, this is once again another DMD mouse. However, this one is transgenic and able to express retinal dystrophin in tissues other than the retina. And you can see the striking appearance in their general appearances just from this image. When we took a look at the lifespan of these mice, all 30 of the DMD mice had actually passed away by the mark of 5 months of age, with the majority of them passing away after the 3-month mark. In comparison to that, our transgenic DMD mice expressing that retinal dystrophin had lifespans that paralleled that of our control mice. I mentioned the fact that abnormal spinal curvatures are very characteristic of DMD, particularly when that muscle degeneration starts to occur in the paraspinal muscles. These are 2 lateral plain film X-ray images. On the right, we have our DMD mouse, and you can see the clearly exaggerated kyphosis of the spine. In comparison to that, the transgenic DMD mouse has a spinal curvature that you would expect to see in a normal, healthy mouse model. These are simply some immunohistochemical studies. On the right, we have, these are cross-sections of soleus muscle. On the right, we have a DMD mouse, and you can see the lack of fluorescence here. That's simply indicating that neither isoform is expressed, full-length dystrophin or retinal dystrophin. On the left, we have a Dmd Transgenic mice, and not only are we expressing retinal dystrophin, but you can also see it's localizing to the periphery of these myocytes. That's indicative of the fact that it is assuming the role of full-length dystrophin protein if it was to be expressed in these DMD model mice. Then we get to my favorite part, which is the heart. So these are cardiac histology slides. On the right, we have the heart of a DMD mouse, and you can see we have the left ventricle here. You can see the clear ventricular wall thinning. This blue staining here is also indicative of fibrosis. So that left ventricular wall thinning as well as the fibrosis would both be indicative findings of some form of cardiomyopathy. In comparison to that, on the left, we have another DMD heart, this one being transgenic with that retinal dystrophin. You can see the stark difference. There's no fibrosis in this example, and you have a very healthy myocardium. The most important finding during these studies when Dr. White was doing them was that of all the DMD transgenic mice, none of them ever reached this point of this cardiac histology slide. Now we get to do the work that Megan and I ultimately did this past summer. So as you can see on the past slides, there was a beneficial attenuation of cardiomyopathy in those DMD transgenic mice. So our job was to now take those images and tie them to the fact that retinal dystrophin was the cause and it was actually being expressed in the hearts of those healthy-looking mice. In order to do that, we turned to the use of a Western blot analysis. So we took protein extraction from heart samples, and then we were going to test for both isoforms, the endogenously produced DP427 full-length protein, and then also human DP260 retinal dystrophin, which would be driven by the transgene. One important note to remember when we're doing these Western blots is that the antibody, the primary antibody that we use here, is specific for the C terminus of the dystrophin protein. The reason why that's significant is because of the fact that that C terminus domain is shared by both the full-length dystrophin isoform as well as the retinal dystrophin isoform. So we can pick up both isoforms with one single primary antibody. We began with looking at the hearts of our control mice. So on the right, we can see we have a black 6 wild-type mice, and when we do the protein extraction from their heart, and we probe for dystrophin, we get a single band right here, which is indicative of DP427, full-length dystrophin protein. That's what you would expect to see in hopefully my heart and your guys' hearts as well. On the left, then, we have a transgenic black 6 mice. Once again, we see DP427, the full-length dystrophin protein, but now we're also picking up another band just above the 250 kilodalton marker, and that is DP260, retinal dystrophin, that protein that is normally expressed only in the eye, now being expressed in the hearts of our mice. Next, we turn to our experimental models, our Dmd transgenic mice and our Dmd mice. As before, when we take the protein extractions from cardiac tissue and then probe for dystrophin, we have our black 6 mouse, as I showed before. We have a single isoform at DP427, the full-length dystrophin protein. Our transgenic mice has the DP427 isoform, as well as a new isoform, that DP260 retinal dystrophin expression in the heart. And then we get to the DMD transgenic heart. And you remember, just take a moment and step back, that healthy myocardium picture that we showed earlier. As it is a DMD mouse, we're lacking the ability to express DP427 full-length isoform. However, we are now expressing DP260 retinal dystrophin in the hearts of these mice. So DP260 protein expression, healthy myocardium. On the other side of that, we have the heart of a DMD mouse. And in this mouse, we pick up no signal. We have the inability to produce DP427 because it is a DMD model mouse, and we're lacking the ability to express DP260 because it's not normally expressed in the heart. However, we wanted to be sure that the reason we were not picking up a signal in that DMD lane was truly because neither of those isoforms are expressed, and not because something was wrong with the sample or that there wasn't any protein in that lane. And so to do that, we turned to the use of a positive control protein, and that was vinculin. Vinculin is another structural protein. It's involved in mediating cell-to-matrix adhesions, and it is also, of course, expressed in the heart. It comes in at a molecular weight of about 117 kilodaltons. And so when we go back to the hearts of our black 6 transgene mice and we use an anti-vinculin antibody, we pick up a band that comes in just above the 95 kilodalton and below the 130 kilodalton molecular weight marker. So that is indeed indicative of vinculin expression. When we turn back to our control models, we run for vinculin, and we do indeed pick up a signal in all 4 protein extracts. So this is once again just confirming the fact that the reason we're not getting any signal in the DMD model is because of the fact that neither isoforms are expressed. Whereas with that healthy myocardium that we saw earlier, we have expression of DP260. So in conclusion, we'll get you guys to lunch. Dr. White's previous work showed that expression of DP260 modifies the DMD mouse from a lethal and severe phenotype to a very mild dystrophy phenotype and gives them a lifespan that parallels that of the control mice. Working with Megan Woods, we were able to show that DP260 retinal dystrophin protein expression in cardiac tissue indeed correlates with the beneficial attenuation of cardiomyopathy. Last thing that I want to reiterate is that, if you guys can remember, about a third of these DMD patients are able and are actively expressing this retinal dystrophin isoform in their eye. So the long-term goal of this project would be to have those patients express retinal dystrophin not only in their eye, but in their muscle, and hopefully reproduce the results that we were showing today. The next step of that would be to identify the human retinal dystrophin promoter and then find compounds that would hopefully induce its expression in muscle. And that is the work of Megan Woods and her thesis. I'd like to take this moment to thank Dr. Robert White. He was my dean in the College of Biosciences. He was also my advisor. He's been a great mentor through this research project. A couple members of his lab, Megan Woods is a co-author. Amber Wiggins McDaniel is a research assistant in the lab, always a great resource and a very positive presence. Bo Wakefield is another graduate student who was working on the mouse retinal dystrophin promoter. I'd like to thank the KCU Office of Research specifically for their support of the SSRF program, and then Kansas City Area Life Sciences Institute for their grant that funds this research, and then lastly, Dr. Paul and Mary Dibidall for their support as well. And so with that, I'd like to open it up for some questions.
We do have time for questions.
Yes, sir.
Again, thank you. Great talk.
Yeah.
Pretty impressive. I was curious if you guys were going to sometime in the future look at some sort of functional studies, either in the gastroc, see if forces are similar to just wild type with the transgenic mouse.
Yeah, and there actually were some relative studies that were published a while back, I think in 2012, is that correct?
Yes.
They weren't necessarily related to the force, but they were related to activity. And it was shown that the DMD mice had a much reduced level of activity, whereas the DMD transgenic mice expressing that retinal dystrophin were equal to their normal wild-type mice.
Cool. And then I just had a follow-up. For those cross-sections, it was really pretty...
Of which,
were they consistent?
Which ones? The hearts or?
Yeah, for the hearts. Were they consistent throughout?
Not all of the, if I'm correct, not all of the DMD hearts were this exaggerated, okay. But the main point was that the transgenic mice expressing that retinal dystrophin isoform never reached this point, whereas some of the DMD mice did indeed.
But were the retinal mice consistent in healthy myocardium throughout as well? Okay, awesome.
Can you share with them about the left ventricular mass?
Oh yeah, and then they also found that in the DMD, the hearts of the DMD mice, the ventricles were significantly reduced in their weight, and that was statistically significant. The heart rate in those DMD model mice was also reduced.
Okay, all right, thank you.
Yep. Any other questions? All right. One more.
Just a couple of questions about the experimental details. So what was the antibody raised against?
It was raised for
What
the.
species was it raised in?
Oh, what species? It was raised in mice, and then the secondary antibody was goat anti-mouse.
Commercially available antibody called
Mandra.
Okay. Okay. And then your transgene, did you have a specific enhancer element that drove expression in the...
It was a
cardiac?
muscle creatine kinase promoter.
Okay. Thanks.
Mhm.
Well, I guess you answered my question. So that's very beautiful study and presentation. So this transgenic mice is muscle specific overexpression.
Yeah,
retinol.
overexpression.
I see. And also the gene therapy right now is kind of like in the phase of develop very advanced. So beside to searching for the promoter activator, the activation of the promoter, Dr. White, are you thinking about the gene therapy for this purpose? Like use viral vector for
Right,
gene
whether it was an
therapy? Yes.
adenovirus or
Yeah.
the other option would be exon skipping.
Right.
Do you have anything to say about that?
So the reason why we feel this is important is because when you do gene therapy in Duchenne muscular dystrophy, it's been shown that expression of dystrophin proteins that the body has not seen is immunogenic.
Yeah.
And there was a paper in the New England Journal of Medicine by Jerry Mendel that showed that expression of full-length dystrophin in mice like this induced T-cell mediated immunity. So we feel that this is important because with the osteopathic principle of the body has the answer to the problem, in 30% of patients, they have the machinery to make retinal dystrophin, and we want to induce that expression in the muscle because we think that would be a good way to treat these patients.
Yeah, my comment is like this retina dystrophin could be have better chance because they don't have the immunoresistance.
Yeah,
So in the gene therapy, you may have a better chance to succeed for this treatment. Yeah.
Thank you.
Thanks.
All righty. Well, thank you guys.